For our project, we decided to study Crohn's disease. Specifically, we sought to examine the genes implicated in the pathogenesis of Crohn's disease and their genetic orthologs in the great ape family. My name is Shiv Shah, and I'm joined by Mark Dansu and Sabrina Seifert. Crohn's disease is an autoimmune disease which affects both the small and large intestines in humans. It is primarily caused by an overactive immune response to a bacteria. The genes implicated in causing Crohn's disease are NOT2, ATG16L1, IL23R, and PTPN22. And these are the genes we sought to identify in the great ape family. First, we conducted a literature review to identify which genes are implicated in the pathogenesis of Crohn's disease. We then used the GWAS catalog to determine common variants, or SNPs, that are associated in each of these four genes. And for each of the four genes we studied, we identified one variant based on its p-value. We then performed a BLAST analysis of each of these four genes onto the great ape family. Finally, we used the NCBI genome browser to identify the location of the variant in human genes to direct us to where in the ape gene a potential variant could be possible to emerge to cause Crohn's disease.
So here are the data. So first of all, we have the distribution of the top BLAST hits on subject sequences. And so we have these red lines, and those represent the subject sequences. And the top line that is in blue represents the query sequence. So from top to bottom, we have gorilla, 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 pan paniscus, pan troglodytes, and pongo abeli. So for NOD2, we see that we have all of these have alignment scores of around 200, which is really great. So really, really close alignment. And for NOD2, around bases 3001 to 3003, we see that this variant, where this variant would take place, the alignment scores are really good on NOD2. When we look at ATG16L1, you look at the variant on this gene, we see that the sequence is also very strongly aligned, except for the sequence in pongo abeli, which is significant. And then looking at IL23R, we have the variant that would take place around base 1259 in the query. And we see that around this space, the sequence is also very strongly aligned. And then likewise in the last gene, PTPN22, we have this variant around base 1908 in the query, and we see that all the sequences are also strongly aligned. Moving on to the average phylogenetic tree. So we made this tree to better picture the relationship between Homo sapiens and the members of the great ape family that we studied. And as you can see, the percent identity for all of these genes studied is very high. We have percents ranging from over 99% to just over 97%, so very close identity. And we see the closest identity in NOD2 and actually throughout all of the genes, but specifically we can look at the species that are most closely related to Homo sapiens, and those tended to have the most percent identity, even though all of the percent identities were very good.